Welcome to Life and Biology. I'm Dr. Joel Graff. This is a condensed lecture for my Principles of Living Systems 100 level biology class. These slides that I'm going to walk through were first presented, were drawn on January 31st, 2022. Recordings on the 4th, so I'm a little behind. Sorry about the tardiness, guys. Anyway, we're talking about proteins, and we had just finished up a little bit talking about nucleic acids, and so this always comes back to the central dogma of biology. I teach this way all the time. And we talk about how DNA can be transcribed to become RNA. Transcribed means that you read DNA and make RNA. Translation is when you read RNA and make protein. And there's also replication, is when you read DNA and make more copies of DNA. On this particular figure, I have two arrows for replication, transcription, and translation, a short one and a long one. The intention here is to point out that each step of the central dogma of biology can be regulated, and that is later on we're going to be learning about the cell cycle and when a cell decides to copy its DNA, how fast cells replicate, the decisions that go on there. We're going to talk about transcription, what factors control the rate of transcription of reading DNA and making RNA. And then there's also features of an RNA that will affect its translation efficiency. And so that's what's going on here, is that we've got RNA being translated to protein at different rates depending on features of the RNA and what RNA binding proteins are in the cell at a given time. Okay, so quick DNA versus RNA. We're not quite done talking about nucleic acids. We will get onto proteins a little bit later this talk. So in nucleic acids, the nucleotides for DNA, to make DNA, are A, C, T, and G are the nitrogenous bases that those have, whereas the nucleotides to make RNA are A, C, U, and G. I underlined the T and the U here. This is uracil, this is thymine. Another difference besides T's are then used when you get to RNA. Another difference between DNA and RNA is this part here where it says deoxyribonucleotides versus ribonucleotides. So deoxy will let you know that there's one less oxygen. So that's what I've got drawn down here, is a deoxyribonucleotide. I don't even know what that says there, because it's 5 carbons. So the reason that I underlined ribo right there means that there's 5 carbons. Yeah, again, when I go from the smartboard to open up my PowerPoint again, I often get some craziness going on with my lines, so that affects the quality here. But the main point is, on a ribose sugar, you have 5 carbons. You've got a 1 carbon, 2 carbon, 3, 4, and 5. So anything connected to the 1 carbon is a 1 prime, and things on the 2 carbon are 2 prime, and 3 carbon are 3 prime, and so on and so forth. We're going to pay attention here that the 3 carbon has a hydroxyl and the 5 carbon has a phosphate. We already talked about that in the last lecture, because we have 5 prime to 3 prime direction. That's the direction that you look down the phosphate sugar pathway as you head off the screen if you were to keep attaching different nucleotides to each other. The nitrogenous bases here, as mentioned above, can be A, C, G, or T. And on the 2 carbon, we have a hydrogen for DNA. Over here, it's pretty much drawn exactly the same, except for my hand, it's all sloppy because of the conversion, but the nitrogenous bases are A, C, G, and U. And on the 2 carbon, there's a hydroxyl on the 2' hydroxyl. And so the difference between this ribose sugar and this ribose sugar is this oxygen sitting right here on the 2 carbon. So whereas you have a hydroxyl on the 2 carbon of RNA, you only have a hydrogen on the 2 carbon of deoxyribonucleotides. Okay. So DNA is often double-stranded, whereas RNA is often single-stranded. So when we draw DNA, it often looks like a ladder, and I'll either draw it on its side or up and down. And we went over the proper orientation to draw 5' and 3' on those in the last video. So anyway, I've got it drawn here. The top strand is 5' on the left, 3' on the right. Bottom strand's 5' at the right, 3' at the left. So they're parallel to each other, but they're running in opposite directions, so that's considered anti-parallel. And because every single nucleotide is matched up with the nucleotide, or every nitrogenous base on the top strand is base pairing with the nitrogenous bases on the bottom strand, it turns into a linear structure. Of course, it goes into a double helix, so it's not exactly linear, but we draw it linearly like this. RNA, in contrast, is single-stranded. So I've drawn a little molecule here. We've got the 5' end of the RNA molecule here and a 3' end here. And so then this RNA, as you go from 5' to 3', follows along this route, comes up, goes off this way, goes around, comes back, goes up, goes around, comes down, and then heads over to the right. And so this is called RNA folding, and it's drawn on a 2D board here, or I drew it on a 2-dimensional object, but these can also form into 3-dimensional shapes. The different parts of the folds have some different names. You can have an RNA where the RNA folds back on itself, and you have double-stranded RNA for a section. That's called a stem. And when you have one stem, it goes around and it comes back the other direction on the other side of the stem. The area where you're going around the circle is called a loop. And then another type of single-stranded RNA structure is I wrote loop here, and that is wrong. That is called a bulge. I should have called it a bulge in class, so I'll have to go back and correct that. But that's a bulge. And then I drew an RNA just straight here, just because I wanted to make a point about nucleotides. You can have the nitrogenous bases pointing down or up, but we still have 5' on the left, 3' on the right. And when I'm drawing the bases up, I wanted to point out that we can take 3 nucleotides here, another 3 nucleotides next to it, 3 nucleotides next to it, and 3 nucleotides next to it. And each time you have a set of 3 nucleotides of an RNA, it will be read and decoded into amino acid language. And so for every 3 nucleotides, there's 1 amino acid. And so RNA to protein is a coded language. It's called the genetic code, and we're going to talk about that here in a second. And there are 3 RNA nucleotides for every 1 amino acid that is coded in a protein. So let's take a look at the genetic code. So this is kind of a sloppy slide at this point, but imagine this slide without any of the writing on it, and you can see that there's every combination of U, C, A, and G that you can have is written here. So again, the genetic code means that 3 nucleotides, A's, C's, G's, and U's, will equal a codon. So 3 together make a codon, and the codon is called, or encodes 1 amino acid. You have a first position, a second position, and a third position. It can be A, C, G, or U at each of those positions. And so there's lots of different combinations. In fact, down here I show the math. If you have 4 options and you've got 3 things in a row where you had 4 possible options, it's 4 to the third is the math behind that. So there are 64 codons. So when we look over here at the genetic code, there are 64 codons shown on here. So UUU is 1 of 64. UUC is another of the 64. I gave the example in class. I just randomly picked U, C, and G, U being the first position. So we see first letter here, and it's a U. C was the second position. So here's the C, so we have to go over to this box. And then G was the third position, third letter, and that was shown here. And I drew an arrow over. So UCG, just randomly picked, it encodes for an amino acid designated SER. That's short for serine. The 1-letter code for serine is S. The 3-letter code for serine is SER. So you'll find amino acid sequences or protein sequences written with either a single letter or a 3-letter code for each amino acid, referring back to the actual name of it. I did a second time. I said, what if it's AAA? And so we found A in the first letter, A in the second letter. We end up at this box by aligning those two up. And then the third letter was A, so we need to take the third one down, and there it is, AAA. So you can really just search for them if you want, but it's faster to pay attention to here and at least find the box first. So anyway, AAA encodes for an amino acid called lysine. Well, amino acid lysine, LYS, that's not a T, that's an L. The drawing just gets messed up when it goes from the smartboard to a PowerPoint. But the 1-letter code for that is a K. Well, why do we have K? It was because L was already taken. We have leucine here. L is for leucine. So once they got to lysine, they had to use a different letter. Why it's K, I don't know. I'm sure you can Google it if you want to find out. There was some other, I went through 2 examples of taking a codon, UCG or AAA, and figuring out which amino acid that it's coding for. So if I were to have this chart up on the board and on a quiz I was giving you a codon, I would expect you to be able to find the amino acid that is encoded by that codon, just to show that you can decode a codon into an amino acid. And then finally, I changed to the black colored marker, and I wanted to point out this particular codon and amino acid. And when you go to do translation, the enzyme that translates RNA into protein is called a ribosome, and the ribosome will assemble fully assemble at the start codon, the AUG. So proteins coding sequences almost always start with methionine. So this is known as the start codon. You can have methionine somewhere within the middle of your gene. It's not considered start there, but you can have methionine throughout your protein. But it's just the very beginning, the end terminus of your protein is going to start as a methionine more often than not. Then we have 3 different stop codons. And so with 3 stop codons, UAA, UAG, and UGA, there is actually not a molecule that'll add any amino acids if this codon ends up being shown, ends up showing up in the sequence. Instead, there's something called a release factor that will bind to these codons, and that is no amino acids, so then you can't keep growing your amino acid chain, and the protein will be released from the ribosome, and then now you've got a free protein, and the ribosome will dissociate and reassociate somewhere else to start making a new protein. Okay, so again, you have a specific codon called a start codon, and you have 3 different ones called stop codons, and that's the start and stop of your protein. Okay, so of those 20 amino acids that we learned about over here, I wanted to talk about their properties. Okay, when you look at these is 10 out of the 20 amino acids are more or less non-polar, and by being non-polar, it means, just like when we learned about methane, the carbon and hydrogen have about equal levels of electronegativity. So there is no real high level of positive or negative charge anywhere on a methane molecule. And so it's, without having any charges, it doesn't want to interact with water. So these nonpolar things are going to be also hydrophobic. If you look at these 10 amino acids, you'll see that everything above the yellow square that I drew a square around, the colored square that I outlined here, you'll see that there's an amino group, a charged amino group, a central alpha carbon, a hydrogen pointing up, and a carboxyl pointing to the right. And it's also the ionic form of a carboxyl group. And again here, same drawing, same drawing. So if you look at all of these, they all have the same thing above the where the yellow is painted. There is one kind of exception here, and that is proline, because proline has a carbon and hydrogen chain that loops around and links back to the amino group on that left side. So proline's a special case. It does differ above the yellow box a little bit, but the rest of them, the yellow boxes, if we go through and look at those, you can see they're almost entirely carbons and hydrogens. We do have a sulfur here and a nitrogen here, but for the most part, out of these large side chains down here, they're going to be carbons and hydrogens. And when you have the short side chains, they're mostly carbons and hydrogens as well. And then here's a cysteine, which has a sulfhydryl group. Cysteine's kind of a special case itself, but anyway, the main thing I wanted to point out is that there's a lot of different amino acids that are non-polar, and when you get a three-dimensional protein, and they all have unique shapes, each protein has a unique shape. So just to draw a protein, I oftentimes just draw a circle, and just know that this circle for the protein could be anything, could be lots and lots of different shapes, but anyway, in the core of the protein, so there's going to be some amino acids that are kind of guarded from water versus interacting with water, like the surface ones. The non-polar amino acids will be in the core, whereas the polar will be around the outside of the protein. That's kind of your first general rule for how proteins fold up into three-dimensional spaces. Of course, it's not a concrete rule. There's always exceptions to the rule, but a lot of times non-polar amino acids will get sunk into a protein that's just floating around in water or cytosol or extracellular fluid or whatever. Okay, if we have non-polar amino acids, then it's not too surprising we have polar amino acids. And just to remember, polar amino acids or polar molecules have areas where there's a partial positive charge or a partial negative charge. Water's a great example. The oxygen is busy stealing the electrons from the hydrogens, and so oxygen is very high electronegativity. So you're going to have, on a water molecule, negative charge around the oxygen. Here we've got a bunch of OHs, which you can find that group on a water molecule, and then sitting next to oxygen on the periodic table is nitrogen, so it is also in the upper right corner where you're going to have some pretty high electronegativity, and so then that's how we have a polar charge there. But then we've got the oxygen as well, so all of these have an oxygen in the side chain. So if you see an oxygen in the side chain, you're more than likely dealing with a polar molecule. The exception to that would be if it's a carboxyl group, you would have actually under physiological pH, the side group would have a negative charge, -1 charge. Over here, we've got some amino acids that would have a positive charge. So here's some amino acids here's an amino group that is the ionic form. Here's another amino group. It's ionic form. It's kind of different than this one I showed here. Maybe it's not even called amino, but here's an amino group here and here, and this one has the positive charge. So anyway, we've got amino acids that can have a full positive charge or a full negative charge, and if they interact within a protein that's folding up or within two proteins interacting with each other, you can get ionic bonds being formed as well. So we have 20 amino acids to keep track of. However, we're going to just group them into 4 groups: nonpolar, polar, positive charge, and negative charge, rather than worrying about all the details. But you can just remember, if you remember your functional groups, then you can remember which ones would be charged, which ones would be polar, and which ones would be nonpolar. Okay, and then to finish off that, no, I've got 2 slides left. So translation is when you take an RNA, you read 3 nucleotides of RNA, and there is an enzyme called a ribosome that will read those 3 nucleotides and will attach 1 amino acid to the growing protein chain. Ribosomes are very large protein structures, and they have two subunits. You've got a large ribosomal subunit and a small ribosomal subunit. Within each, the ribosome will span a length of RNA such that there are 3 nucleotides towards the front end, if you want to call it that, the middle and the back end of the ribosome. And why is this the front end? Is because ribosomes move in a 5' to 3' direction. So this particular RNA would be moving to the right. And if it moves to the right, it's going to actually jump to the next 3 nucleotides, so that these 3 nucleotides that were here are then here. And then the next time it jumps 3 to the right, the 3 nucleotides that were here and had come here are then going to be here. So it's a constant moving forward 3 nucleotides at a time, and that shifts which site the nucleotide or the codon is within the ribosome. So we have got 3 different sites. The one furthest towards the 3' end is called the A site, and this is where a tRNA is going to come in. The tRNA will be holding on to 1 amino acid. It's going to be right here in the A site, which is next to the P site. A is for adding in, I think. P is for polypeptide, and so you've got a polypeptide chain. There's a tRNA interacting here, and it's got the growing polypeptide chain on the top of it. I'll draw this out a little different on the next slide so you can kind of understand. Between this slide and the next slide, we kind of cover the idea of translation, and then in class we watched a animation. And then we've got the E site after that. Well, what happens is that the growing polypeptide chain that was on the P site gets transferred over to the amino acid that was in the A site. That means this amino acid doesn't have this tRNA doesn't have an amino acid anymore. And when the whole ribosome jumps forward by 3, the tRNA that was in the P site now is in the E site, and it can exit. This is where since it doesn't have an amino acid anymore, it doesn't have anything to contribute to the growing protein, it can just be released. Okay? And then it'll just keep shifting to the right. So I made a little point here where we're going to zoom out so that we could see, so far that we can't necessarily see individual nucleotides, but this red line is the RNA. It's going from the 5' end to the 3' end. We've got a start codon here, so this is the start site for translation. We've got a stop codon here, so this is the stop site for translation. And then we've got 3 different ribosomes. Here's the large subunit, small subunit, large subunit, small subunit, large subunit, small subunit. So those are moving along, and they're moving from a 5' end to the 3' end on the single-stranded RNA, which is called the messenger RNA. So we're zoomed out, and if a ribosome is on an RNA, if more than, oh man, you can't even read this. If more than one ribosome is on an RNA and they're all actively translating, it's known as a polysome. So here we've got 3, so this would be considered a polysome with 3 ribosomes on it. The ribosome that is closest to the stop site started way over here, and it's been adding amino acid for every 3 nucleotides it moves. So it's a fairly long polypeptide chain, and it's folding into a 3-dimensional structure over here. The one that was closest to the start site, it's going to have the shortest polypeptide because it has not read as many codons, the 3 nucleotide codons, and so it doesn't have as many amino acids on its growing. And then, of course, if a ribosome is somewhere between those extremes, it's going to have a medium length polypeptide growing out of it. Okay, and that was it for that lecture. So there's a lot of drawing going on, but now that we've kind of gone through translation, then we're going to talk about protein structure.